Good day everyone. Today our group will be presenting on Genome-wide Association Studies or GWAS. My name is Celine and I will start with the general workflow of GWAS. GWAS follow a three-stage workflow. First, in the pre-GWAS stage, we prepare the data by designing the studies and assembling genotypes and phenotypes datasets. We then perform initial quality control to filter out SNPs, missingness, and so on. After that, we then correct for population stratification using PCA. These steps ensure our data is clean and unbiased. Second, in the GWAS stage, we run the actual association test. Each SNP is tested using the regression models, which linear for continuous threads and logistic for binary threads. We then apply multiple testing corrections like Bofferoni or FTR to control the false positives. Finally, we interpret results using summary statistics, Manhattans, and QQ plots. Third, in the post-GWAS stage, we move to downstream analysis such as polygenic risk scores, pathway analysis, or functional annotations. This helps us to connect genetic signals to the biology and predictions. Moving on to the quality control in the GWAS. It is a critical step in GWAS because poor data can lead to false association. We filter individuals and SNPs based on missingness, remove rare variants with very low minor allele frequency, and exclude SNPs that deviate from Hardy-Weinberg equilibrium. We also check heterozygosity to detect contamination or inbreeding, and also remove the duplicates or closely related individuals to avoid bias. Additional steps like LD pruning for independence, sex checks, and testing for non-random missingness. Together, this QC filter ensures that only high-quality, reliable data enter the GWAS pipeline. Now, we will look at the principal component analysis, which is PCA. PCA has four main objectives. First, it reduces the dimensionality by shrinking thousands of SNPs into a smaller set of variables while keeping the essential patterns. Second, it removes redundancy by turning correlate features into independent principal components. Third, it improves efficiency by speeding up computation and making visualization easier. And finally, it highlights the variance by capturing the direction in which the data varies the most. Together, this aim makes the PCA a powerful tool for correcting population stratification in GWAS.
On this slide, we summarize the major steps of PCA in GWAS. First, we perform SNP filtering to remove the low-quality variant. Then, we do LD pruning and exclude the related individuals to ensure independence among samples and SNPs. Next, we run the PCA on this LD prune and related SNPs to capture the main axis of genetic variations. After that, we project all individuals onto the PCA space, which allows us to visualize the population structure. Finally, the results are ready for visualization, helping us to detect antic outliers or substructure. Tools commonly used in the PCA include Blink and the reference panels like the 1000 Genome Project.
Hi, my name is Ethan. My groupmate, Celine, just now has explained the theoretical part of our workflow. So, now I will demonstrate how I actually perform these steps practically using the computer. For this analysis, I utilized the 1000 Genome Project Phase 3 dataset aligned to the HG19 genome build. Specifically, I analyzed 504 East Asian individuals across 5 distinct populations. And I processed this data on a Linux system using a tool called Blink. The first step is quality control, which you can see in the first block of the code. I applied split filters to clean the raw dataset. Then, I used the MLF 0.01 to remove the rare variants that appear in less than 1% of the population. And I used the Genome 0.02 and MIND 0.02 to exclude any markers or individuals that were missing more than 2% of their data. And most importantly, I used the command remove to filter out samples that had abnormal heterozygosity, which usually indicate contamination. This can ensure our data is high quality before we start. Moving to the second block of code, we focus on removing the bias using the Blink 2.0. There are several key parameters here. Here, I set a trace tool to speed up the calculation and reapply MLF 0.01 to strictly ignore the rare variants during this step. Then, I use the exclude flag to remove known high LD regions that can confuse the analysis. Then, I ran in-depth pairwise to prune the remaining markers so they are statistically independent. Then, I also used the command king cutoff 0.0884 This identifies and removes the people who are closely related, specifically up to the second degree like the brothers or cousins. And the reason we are doing this is because it guarantees that our final plot shows the real population history rather than just a family tree. Once the data was clean and is unrelated, I move on to the calculation using the Plain 2.0. And as you can see in the third code block, I use the keep and extract command to tell the program only to use the clean list of people and variants we created in the previous step. And then I calculated the top 10 principal components. Finally, I used the RStudio to visualize the result, and the plot confirmed that our QC filter successfully preserved the biological signal. First, let's see on the right side of the plot, we can see that the Japanese group in the purple color form a tight, completely separate cluster. This clear gap indicates that distinct genetic isolation compared to the other group. Second, in the center of the plot, we can observe a continuous linear gradient connecting the northern Han in blue color and the southern Han in green color. Rather than splitting into two separate blobs, they merge into each other, which represent a smooth genetic transition from north to south. Finally, on the far left of the plot, the Dai in red color and Vietnamese in grey samples overlap and are clustered tightly together. This indicates that they share very close ancestry. And in conclusion, because we carefully clean the data and remove the relative that are closely related, we can see the population groups clearly. And we will now use this PCA result to adjust for ancestry differences in the final analysis. And my groupmate Celine will now continue to explain how this works in the regression model for GWAS.
Alright, let's look at the association analysis. In GWAS, we use regression model to test whether a genetic variant or SNP is associated with a trait while adjusting for other factors. There are two main types of regression models used. Linear regression is used when the trait is continuous, such as height or MBI, while the logistic regression is used when the trait is binary, for example disease versus control. Although these two models are used for different types of traits, the variables included in the models are the same. In both cases, the SNP genotype is the main variable of interest, usually called as 0, 1, or 2 copies of the effect allele. We also include covariants such as age, sex, and principal components from PCA to control for population structure and other confounding factors. The outcome we are studying is the phenotypes. The key difference is how the effect is interpreted. Linear regression estimates the change in trait value, while the logistic regression estimates the change in disease risk. But, conceptually, both models answer the same question, which is whether a SNP is associated with the trait after adjusting for covariance. After running the regression for each SNP, GWAS produces a summary statistics file. GWAS summary statistics contain many standardized headers. However, these headers can be broadly grouped into three categories, which are variant identification, allele information, and association statistics. Variant identification tells us where the SNP is in the genome, while the allele information specifies which allele is tested. Association statistics describe the effect size and statistical significance. These core headers are sufficient to interpret GWAS results and generate the Manhattan and QQ plot.
This is a Manhattan plot showing the GWAS results for our traits across all 22 autosomes. Each dot represents a single SNP, plotted by its genomics position on the x-axis and the negative log 10 p-value on the y-axis. The higher the dot, the stronger the statistical association with the trait. The dashed line indicates the genome-wide significant threshold, which is generally set as 5 times 10 power to the negative 8. Any SNP above this line is considered significantly associated with the trait after correcting for multiple testing. Here we see several peaks surpassing the significant threshold. For example, SNPs on chromosomes 1, 2, 7, and 20 show the strongest associations. These regions are potential candidates loci for further investigations. Each peak represents not just one SNP, but a cluster, due to linkage disequilibrium among nearby variations. Most other SNPs fall below the threshold, which is expected because most variants has no strong effect on the traits. These significant regions are potential candidates loci for further investigation and downstream analysis. This QQ plot compares the observed versus expected p-values from our GWAS. On the x-axis are the expected value under the null hypothesis, while the y-axis show the observed values. The diagonal lines represent the perfect agreement with the null. Most points closely follow this line, indicating that our analysis is well controlled for confounding factors such as population structure. At the upper right tail, points deviate strongly upwards, reflecting the SNPs that are significantly associated with the traits. The genomic control factor is 0.99, very close to 1, which further confirms that there is no inflection in our GWAS statistics. In short, the QQ plot validates the quality of our GWAS and supports the significant association seen in the Manhattan plot.
Hi, I'm Jiwei. I will discuss the post-GWAS analysis. There are several types of post-GWAS analysis, each with a different purpose. Functional annotation helps to assign biological meaning to the variants. SNP heritability, estimation, and LD score regression measure the overall genetic contribution to the trait and help detect confounding effect. Gene or gene set tests identify genes and biological pathways associated with the trait. Fine mapping helps pinpoint potential causal variants, while polygenic risk score estimate overall genetic risk. Other analyses, such as Mendelian randomization, colocalization, and GWAS, are used to study causal relationships and link genetic variants to gene expression. In this study, the post-GWAS analysis we performed were functional annotation, SNP heritability estimation, LD score regression, and gene or gene set tests. Due to time constraints, we will focus only on functional annotation and LD score regression. First, let us look at the functional annotation. We perform the functional annotation using a software tool called ANOVA to assign biological meaning to variants. It tells us the physical locations of the variants and predict their effect on genes. This table shows the result of our functional annotation. These 5 columns are the key features of the annotation's result. The most important feature is FUNCREF gene, which indicates where each variant is located relative to gene structure. Upstream and downstream indicate that the variant is located before the start and after the end of a gene respectively. Intergenic means the variant is located between genes. Intronic indicates that the variant is located within a gene but outside the coding region, while exonic means the variant is located within the coding region of a gene. Values starting with ncRNA indicate that the variant is located within a non-coding RNA gene. Gene-ref gene shows the gene name associated with one variant. UTR3 or 5 means the variant is located in a 3' or 5' untranslated region of a gene. Gene-ref gene shows the gene name associated with one variant. Gene detail ref gene indicates the physical distance between the variant and the nearest gene. It is only filled in when the variant is not inside a gene. Exonic FUNCREF gene shows functional consequences if the variant is exonic. In our result, exonic variants are classified as either synonymous or non-synonymous single nucleotide variant (SNV). A synonymous SNV is a single nucleotide change that does not alter the amino acid sequence, whereas a non-synonymous SNV causes an amino acid change. AA change ref gene provides the specific amino acid change caused by the variant. Other features, such as CHR, start, and ref, and ALT indicate chromosome number, start position, end position, reference base, and alternate non-reference alleles occur on at least one of the samples. There are several interpretations that can be made based on the result. Firstly, the majority of GWAS-associated variants are located in non-coding regions, particularly intronic and intergenic regions. This suggests that most genetic signals are likely acting through regulatory mechanisms, such as affecting genes expression, rather than directly altering protein sequences. Secondly, at the regulatory level, many variants are located upstream or downstream of genes, where they may directly influence gene expression rather than protein sequence. For example, variants located upstream and downstream of MIR12136 may affect the expressions of this microRNA, potentially impacting post-transcriptional gene regulation. Thirdly, when focusing on variants with direct protein-level consequences, the gene MEGF6 stands out. MEGF6 contains the majority of the non-synonymous SNV, including multiple missense variants as well as intronic variants. The clustering of both coding and non-coding variants suggests that MEGF6 may be an important locus associated with the trait. In conclusion, the results suggest that most genetic signals act through regulatory mechanisms, while a smaller but important subset of variants directly alter protein function. Although the majority of GWAS-associated variants are non-coding, we identify high-confidence coding variants in MEGF6, which represent strong biological candidates for further functional validation. Now, let us move on to LD score regression. We perform the LD score regression using another software tool called LDSC to estimate SNP-based heritability, which is the proportions of trait variation explained by SNP and detect confounding effect using only summary statistics. This picture shows the result of our LD score regression with metrics including total observer skill H-square, lambda GC, mean chi-square, intercept, and ratio. Total observed skill H-square is the estimate of SNP-based heritability, the proportions of trait variance explained by SNP used in the analysis. Lambda GC shows overall inflections of test statistics, or how much the p-values deviate from what we would expect by chance. Mean chi-square shows the average range of association across all SNP. Intercept estimate confounding, such as population structure of relatedness. Ratio tells us what proportions of the inflection in test statistic is due to the confounding rather than true genetic signal. Based on the result, several interpretations can be made. Firstly, the result is not reliable as the total observed skill H-square is greater than 1, which is biologically impossible. This may be due to noisy GWAS summary statistics and low signal-to-noise ratio, resulting in an unstable heritability estimate. Secondly, no inflation is observed. Both the lambda GC and mean chi-square values are slightly below 1, indicating that the test statistics are deflated rather than inflated. This suggests that the GWAS is underpowered, with weak or no true genetic association signal detected. Thirdly, the results show no confounding effect as reflected by the intercept value below 1. Since there is no inflection, the ratio value is not applicable and cannot be used to estimate confounding contributions. In conclusion, the LD score regression result show no inflection or confounding, but the GWAS appears underpowered, leading to conservative test statistic and an unreliable SNB heritability estimate.
In conclusion, our assignment shows that GWAS is essential for connecting genetics to the traits. It is also important to note that rigorous data cleaning is the key to minimize the bias and to ensure reliable results. Moving forward, the goal is to go beyond just statistics. For example, we can validate these findings in the lab to understand the actual biological mechanism other than just doing GWAS. This is important because if we understand how the genes work, we can eventually use that knowledge to predict disease risk and design better treatments. And that's all from us. Thank you.